Okay, so blood typing, human blood groups and such. Your red blood cells on their surface membrane have proteins that they can express, like all cells do. And these have different types of proteins. Sometimes they will express this A, sometimes they'll express this B. These are your agglutinogens. And you can have the agglutinogen labeled A, or you can have the agglutinogen labeled B, or you can have zero, none. And that would be the agglutinogen 0, but we don't say 0, we just say O. There's another one, the RH, the rhesus monkey factor. That's what it's referred to as. And you either have that agglutinogen, that RH agglutinogen, you are RH positive, or you don't. You are RH negative. And all. So when we look there, you'll notice that there's types of blood that are out there. And then where did you get it from? You get it from your parents. And so when you look at the genes, the A/A right there, that says you received an A gene from one parent and an A gene from the other. So A gene from mom and an A gene from dad. If both of your parents are type A, they could have been A or they could have been AB. If both of your parents are type A, you're not going to be type B. If your parent was type O, they have neither and they give neither. So so if one parent is type O and the other one is type A, chances are you're going to be type A. Or type O, 50-50 on that one right there. So, but you can see the blood groups, the percentage of peoples. Most people, 40 to 50% are type O, and then the other big chunk is type A. Type B is kind of more rare, and type AB is very rare. The antibodies. So if you have type A, you have these antibodies which will attack type B. If type B blood shows up in you, your body will attack it. So you can't receive type B blood. You can receive type A blood, or you could receive type O, because O has no surface antigens. Same thing with the B. The type AB, if you're type AB, you don't have any antibodies. So you can actually receive blood from any other blood type. If you're type O, you've got antibodies for both A and B. So you can only, if you're type O, you can only receive blood of type O. Agglutinins, these are the antibodies and such. So the agglutinogens is the surface protein, and the glutenins is the antibody. That's kind of, sometimes that confuses people because they sound so similar. Don't let that bother you. When a wrong type shows up, or when the opposite type shows up, you get this clumping, this agglutination, that can then trigger, if you were to be given the wrong type of blood, if a person, a type A, was to be given type B, you would get these clotting forms, these clumps, this agglutination. And over time, that would then cause the red blood cells to rupture. And that releases all of this hemoglobin, all this protein, these big proteins and such. They then go through the kidneys and they kind of clog up the kidneys and they damage the kidneys. So if you were to receive the wrong blood type, and if they catch it in time, they try their best to dilute it out and such, and then they try their best to get you to remove it, to urinate it out. Like I mentioned, the other one's the RH factor. It's a different set. You either have it or you don't. You can have a transfusion reaction. It's just not as bad. It's bad, just not as bad and such. And sometimes mothers and babies, they have different. And also an RH negative mother, she doesn't have it. So she has, if she has an RH positive baby, the first one, at birth, when the placenta separates, then some of baby's RH positive blood mixes with mama's RH negative. And then she will produce these antibodies. It doesn't affect the first baby. It's the second baby. So now she has these antibodies. And so she's RH negative, and she has these antibodies now for RH positive. She has an RH positive baby. Everything's great. At birth, when the placenta ruptures, some of mama's blood mixes with baby's with those antibodies, and it attacks baby's blood. And it causes the baby's blood to clump and to clot, and then you get this erythroblastosis fetalis. To prevent that, they used to then transfuse the baby with mama's blood. Boom, right there on the spot. Because when your newborn baby turns blue, everybody freaks out. Now, no matter what, first baby, 10th baby, they give you a shot of this RhoGAM, and it prevents mama from making the antibodies. So when there is a birth, she doesn't produce any antibodies. She can't pass that along. Everybody's happy. So how you identify blood? The old days, you take the blood, mix it together. If it clumps, don't use it. If it doesn't clump, use it. Now they have these Eldon cards, and these Eldon cards have in these little bitty, they have 4 circles, and the first one is A. It contains the anti-A. And the second one is B. It contains the anti-B. And the third one, the D, it contains the anti-RH. And then the fourth one is a control. It doesn't have anything in it, nothing. So you add a drop of blood to all 4 of those. You mix them up. You let it set. And then you compare A to the control, or you compare B to the control, or you compare the D to the control. And you look for clumping. If it clumps, that antigen or that agglutinin is there. That agglutinin is there, so it has the type A. Or if it clumps in the B, it has the type B. If it clumps in both, it's AB. If it doesn't clump in either A nor B, it's type O. Then you look at the third one, the RH. Either it clumps or it doesn't. If it clumps, it's Rh positive. If it doesn't, it's Rh negative. And so then you just do it and you read the card and you can tell what type of blood it is. Pretty easy. No. All right. So what is shock? Shock is the loss of blood. And then the body has a whole problem with that and such. There's different types of shock and such. But what you try to do when there is a rapid blood loss for one reason or another, you try to increase blood volume. When you lose blood volume, you lose blood pressure. Your internal organs work off of blood pressure, because without pressure, you can't get the appropriate nutrients and waste, the appropriate nutrients in, the appropriate waste out. Bad things happen, everything shuts down. So you can use just pure plasma. They can add things which will cause your plasma to expand, these plasma expanders and such, or you can just use saline solution. Most of the time when you're getting fluids in the hospital, it's saline solution. So different diagnostic tests that can be run on the blood. You can look for anemia. You remember a normal hematocrit was 45. Below that, you have anemia. If the plasma is really yellow, you have a lot of fat in there. You can measure glucose levels and such, look for diabetes. You can then do if your white blood cell is elevated, then you have an infection. Then you would do a differential count to see what type of infection. They'll do a clotting time on it as well. They'll measure your platelets and such. Basically, you go in and they'll order a complete blood count and just get it all done at one time. So, all right, there you go, blood.